Some understanding of the proposed evolutionary origins of and relationships between ferns provides back-of-the-mind knowledge helpful when attempting to identify them. How our three Polypodies came to be has been persuasively and confidently established. Thanks to Professor Irene Manton and her students at Leeds University in the 1960s, plus recent work carried out in the USA, the explanation of the diversity within the Polypodies is now well known. In Britain we have a triangle of 3 species and 3 sterile hybrids between them. The southern Polypody, Polypodium cambricum, is, as far as our knowledge goes back, the original European ancestral species. It has the base 2 sets of chromosomes. Our common Polypody, Polypodium vulgare, has proved to be the result long ago of hybridization of 2 northern species, Polypodium sibiricum and Polypodium glycyrrhiza. Both have the base 2 sets of chromosomes, but a division failure in gamete production resulted in doubling, so that our Polypodium vulgare has 4 sets. Our third species, the western or intermediate Polypody, Polypodium interjectum, has 6 sets of chromosomes derived by the crossing of the common and southern Polypodies. Its being a distinct species requires a bit of explanation. First, though, the 3 hybrids. One of them, between the common and intermediate Polypodies, is sterile because it has an odd number of chromosome sets, 5. The next hybrid, between the intermediate and the southern Polypodies, is again sterile, though with an even number of chromosome sets, carrying genes which are just too different to allow reproduction. The third hybrid is between the southern and common Polypodies. It is also sterile, with an odd number of chromosome sets, 3. However, this hybrid is different from the others. Sometimes an anomaly occurs that doubles the number of chromosomes, rendering the new Polypody fertile and capable of reproduction and spread. It is a new species. Each iteration of this new intermediate Polypody can hybridize with its parental species, but those hybrids are sterile, for the time being, in evolutionary terms, maintaining the eternal double triangle of Polypodium species and hybrids. What distinguishes our 3 Polypodies? For observations and identifications in the field, you will need a hand lens with at least 8 times magnification. The old magnifying glass with a handle is useless. You might as well just look closer. If you're very short-sighted, try squinting close to without your glasses, as I used to do before cataracts and almost normal vision. Unfortunately, now I'm a bit handicapped. Hold the lens close to your eye and bring the specimen up underneath it until its detail comes into focus. Doing it this way doesn't work. The southern Polypody, so called because Britain is its most northerly European outpost, while it grows in southwestern Britain. The western Polypody is also called the intermediate Polypody because of its sharing characters with its evolutionary parents, the other 2. The common Polypody has been known as that since long before botanists appreciated there were 3 species, and of course it's very common. The only way to be certain which Polypodium species you have is to examine its sporangia, the spore cases which occupy the undersurfaces of fronds, Greek for spora meaning seed plus angion, a vessel or container. Sporangia are aggregated in clusters known as sori, singular sorus, from the Greek soros meaning, very appropriately, a stack, pile or heap. The sori begin small and the same green as the frond surface, enlarging and changing colour as they mature. Polypodium sori lack the indusium other ferns have that protect the sporangia until they are ripe. The shapes of Polypodium sori differ species to species, but are often too difficult to define to be used as an indicator of species, but can be used as just a hint. Thus the sori of P. cambricum and P. interjectum may appear oval, while the sori of P. vulgare may be described as circular, though round is a safer term. As the sori ripen, they tend to coalesce, and their outline shape becomes even more difficult to describe with any certainty. However, used with care, oval versus circular is not an entirely useless consideration. A single sorus consists of a little heap of spore capsules, the sporangia. Each sporangial head is a more or less spherical envelope of cells with a ring around it, the annulus, the purpose of which we will see later. Here are three views of a Polypodium sorus, from above, from the side, and in section where the sporangia are seen to be long-stalked, all attached at the centre of the sorus. To examine them individually, we must scrape them off and spread them on a microscope slide, which may seem brutal, but provides plenty of opportunities to piece together fragments that provide an understanding of sporangial anatomy, with the bonus of a few complete examples, as we see here. Here are some close-up views of sporangia on a microscope slide, taken with a digital microscope costing about £150. Pay £35 and you'll likely get only £35 worth of microscope. And here is what we see with the digital or stereo microscope, left, compared with the compound microscope enhanced using focus stacking software, right. The inset shows the rear view of a sporangium's annulus, seen in profile in the main pictures. Hopefully that brief exploration makes better sense of these views. Now we'll examine the Polypodium sporangium in microscopical detail so that we may count the main diagnostic characters, the numbers of indurated cells in the annulus, and the 1 to 4 basal cells, depending on species. The annulus, a row of cells around the circumference of the capsule, consists of some cells with normal thin walls and 5 to 15 with walls thickened on their inner and adjacent sides. When the sporangium is ripe and the weather warm and dry, water evaporates from the indurated or thick-walled annular cells and their contents shrink. The thin outer walls become concave, while the inner walls are pulled together enough to set up tension along the annulus, and the sporangium splits open on the opposite side where there is a weak spot, and the spores are catapulted away from the frond. Here is a sequence of spore dispersal. A shows the intact sporangium. As the annular cells lose water into the dry atmosphere, the annulus contracts and the capsule splits open, B, C and D. When fully stretched, E, suddenly a little bubble appears simultaneously in all the annular cells, F, releasing the tension and catapulting the spores away from the frond to be caught by the breeze. Here's the way that spore ejection mechanism works, shown as animated diagrams. And again. Field identification of polypodies can be straightforward, but to be confident, some microscopical examination is required. Experience has shown that what seemed to have been a correct identification turned out to be contradicted by the microscope. Please don't be discouraged, just stay realistic. For confirmation of your field identifications, you will now need the whole kit, or as much of it as you can manage. A powerful hand lens will, after a lot of experience, just about enable you to quantify the features used for comparisons, while a reasonably priced digital microscope, don't buy the cheapest, will make that just about possible. For best results you really do need a high-power microscope, but with care you can certainly get a good idea of the number of indurated cells, few, many or intermediate, with a decent hand lens. In the field and without microscopes, Polypodium identification should always be considered provisional, and that can be disappointing, indeed discouraging. Polypodium cambricum can usually be identified confidently, but the other two frequently overlap in appearance, which leads to uncertainty, which should be maintained. After all, they are closely related. That is just an unfortunate biological fact, but perseverance will eventually be rewarded. The first problem is that all three take on various forms according to where they are growing: robust or less so, stiff or floppy, upright or pendant, in the shade or open, on rock, soil or tree. Also, P. interjectum expresses noticeable genetic variability as well. Good luck! Three microscopical characteristics separate the three polypodies: the number of indurated and basal cells, and paraphyses. We'll deal with paraphyses first. Paraphyses, singular paraphysis, are branched sterile hairs that occur among the sporangia, but only in the sori of P. cambricum, which is useful. They are just visible with the hand lens, but are quite sparse, therefore difficult to find. These in the main photograph were imaged with a digital microscope and enlarged. Listing the three characters for each polypody, P. cambricum has 1. Paraphyses present, 2. On average 5 to 10 indurated cells, and 3. 3 to 4 basal cells. P. interjectum has 1. No paraphyses, 2. On average 7 to 9 indurated cells, and 3. 2 to 3 basal cells. P. vulgare has 1. No paraphyses, 2. On average 10 to 14 indurated cells, and 3. Just 1 basal cell, which is the most difficult to see without a compound microscope. The easiest feature to count is the indurated cells, which must be applied to many sporangia to calculate the average, which hopefully will match one of these ranges, as well as your field observations. If you don't get a match, as we have learnt the hard way, the specimen must be designated the tongue-in-cheek taxonomic rank we apply to unidentifiable male ferns, WOB. W-O-B, just walk on by. Confirmatory characters for the southern polypody, Polypodium cambricum. Difficult to see they may be, and there are no paraphyses visible in any of these sori. After a thorough search, I found these paraphyses elsewhere on the frond. The smaller colourless sporangia contain no spores. Some paraphyses photographed on the digital microscope and enlarged, and then on the compound microscope, assisted by focus stacking software. Here are some examples of what we can see when attempting to count the indurated and basal cells. These sporangia show how doubt can prevent confidence, obliging us to assess other sporangia. Here both characters can be clearly seen, but they're not the same sporangium to sporangium. Counts must be repeated with other sporangia so that meaningful averages may be calculated. Here too, confidence is assured, and the one on the right confirms that both cell counts sit within the published averages. Confirmatory characters for the western polypody, Polypodium interjectum. If you catch P. interjectum with fully developed sporangia at their earliest stage, the indurated cells will not be coloured, as they will be in P. vulgare at the same stage. Even here you can detect just the beginnings of darkening, and this character does not last very long. Very soon the indurated cells of both species have darkened, and the rotundity or ovalness of the sori is difficult to be sure of. Identification will be dependent upon microscopical characters. With good specimens of sporangia, counting is possible but must be repeated to obtain a confident average. Here we can tell the difference between the indurated and other cells of the annulus by their outer walls, concave in the shrinking indurated cells. Counts are confident. Confirmatory characters for the common polypody, Polypodium vulgare. The row of indurated cells is clearly seen contrasting with the rest of the sporangium. As ripening progresses, the sporangia increasingly turn yellowish, then golden, and the number of indurated cells may at least be estimated, though they're obscured by adjacent sporangia. To be sure, sporangia need to be teased apart, A tricky but not impossible procedure. Once again, the compound microscope provides the best views for counting. The numerous indurated cells are clearly defined, while the single basal cell is small and can be difficult to see. Here are some alternative views which might not provide a count of indurated cells, but they do clarify the one basal cell condition. I spent a lot of time trying unsuccessfully to film sporangium dehiscence, as I have with great success with other ferns, which shed all of their spores in a matter of 5 minutes or so when coaxed with a little heat from an old-fashioned light bulb. These guys really take their time. See my video, How Ferns Shed Their Spores. The link is in the notes below. With patience, I did see several sporangia pop, but caught only one good example on video. I'll keep trying to get better footage, but when this short clip is done, I'll show you how a male fern does it. Next, at full magnification, a single saurus at 5 times normal speed shown 3 times. Now the whole process becomes visible and, I suggest, unforgettable.